All right, today we are diving deep into the seriously dynamic world of the eukaryotic cell. We're going to go on a journey from its innermost scaffolding all the way to its outermost connections. And along the way, we'll uncover the secrets of how a cell organizes itself, how it moves, and how it interacts with its environment. You know, if you could actually shrink down and step inside a cell, you wouldn't find some big empty space with organelles just floating around. No way. It would look a lot more like this. A super dense, crowded cityscape just packed with structures, membranes, and this incredibly complex web of protein filaments. I mean, at first glance, it just looks like pure chaos. So that really brings us to the central question for this whole explainer. How on earth does a cell manage to not only maintain its shape, but also bring some kind of order to all this chaos? Well, the secret lies in a really elegant and dynamic internal framework. So here's our game plan. We'll kick things off with the cell's internal framework and see how it actually doubles as a highway system. Then, we'll check out how cells move, and finally, we'll zoom out to compare the armor of plant cells with the flexible matrix you find in animal cells before we tie it all together. Okay, so the secret to all this cellular order, it's the cytoskeleton. I want you to think of it as a dynamic network of protein fibers that acts as the cell's skeleton. It provides not just structural support, but it's also this amazing organizational scaffold that holds everything in its proper place and connects it to the outside world. So this cytoskeleton is built from three main types of protein filaments, and each one has a very specialized job. First up, we have the flexible actin filaments, which are just crucial for cell movement and contraction. Then you've got the larger, more rigid microtubules. These guys act as railways for internal transport. And finally, there are the incredibly tough intermediate filaments, which are all about pure structural stability, helping the cell withstand all kinds of mechanical stress. And right here, you can really see the structural difference. On the left, actin filaments are basically like two thin protein chains just twisted together, and that's what gives them their flexibility. Now look at the right. Microtubules are much larger, hollow tubes. Their rigid, pipe-like structure, well, that makes them the perfect tracks for the cell's internal transport system. Okay, so if microtubules are the highways, how does cargo actually get from point A to point B? Well, the cell has a super sophisticated transit system, and it even comes complete with molecular trucks to get the job done. For this system to work, you need four key components. First, you have the cargo, which is usually packaged in a little membrane bubble called a vesicle. Second, you need a motor protein that actually burns energy to create movement. Third, a connector molecule to link the motor and the cargo. And finally, of course, you need the microtubule track itself. And this is what it looks like in action. It's so cool. You can see a motor protein complex. In this case, it's dynein, and it is literally walking step by step along the microtubule, carrying its vesicle. It's just like a tiny autonomous delivery truck driving down a highway, all happening inside the cell. But hey, this isn't some chaotic free-for-all. This highway has traffic lanes and a clear direction. You've got motor proteins called kinesin, and they handle the outbound traffic, moving cargo out towards the cell's edge. We call that the plus end of the microtubule. Meanwhile, dynein handles all the inbound traffic, moving things back toward the cell's center or the minus end. This really strict regulation is what prevents total chaos and makes sure materials get where they need to go efficiently. This internal framework is just incredible, but its role doesn't stop at the cell's edge. The very same components, microtubules and motor proteins, are actually repurposed to power the movement of the entire cell. And they do this by creating these remarkable external structures, like cilia and flagella. The core of both cilia and flagella is built from microtubules, but they're arranged in this very specific, very precise pattern called the 9+2 structure. And this is not a random jumble. This is an elegant piece of molecular machinery that's been conserved across huge stretches of evolutionary time. It just works. Now this cross-section shows that 9+2 arrangement perfectly. See the 9 pairs of microtubules in a circle around 2 single ones in the center? So how does it move? Well, movement is generated by dynein arms. Yep, the same type of motor protein we just saw carrying cargo, which are attached to one pair and then walk along the adjacent pair. This sliding action forces the whole structure to bend, and that creates a really powerful whip-like motion. All right, let's switch gears. We're going to shift our focus from the cell's interior to its exterior, and from movement to protection. We'll start with plants because they build this incredibly rigid protective barrier around themselves, the cell wall. So, when two plant cells form right next to each other, the very first layer they create is called the middle lamella. It's this sticky substance made of pectin that basically acts like cement, gluing the individual cells together to form tissues. As a young plant cell is growing, it first lays down a flexible primary wall. But once it hits its full size, it often reinforces itself by depositing a much stronger and more rigid secondary wall on the inside. It's this layered construction that gives plants their immense structural strength. And this wall isn't just some passive box. Its construction is an active, highly integrated process. If we trace the path, it all starts with genetic instructions in the nucleus. These get sent to organelles like the Golgi apparatus, which acts like a processing and packaging plant for the wall's components. Then, these components are transported to the plasma membrane for final assembly into a complex matrix of cellulose fibers, which, that's what provides the main structural framework. Animal cells, on the other hand, don't have a rigid wall, so they have a completely different strategy. They secrete a complex and dynamic web of proteins and carbohydrates around themselves, and this is what's known as the extracellular matrix, or just the ECM. Here, we can really see how the components of the ECM all work together. You have these strong collagen fibers providing tensile strength, kind of like rebar in concrete, and they're embedded in this gel-like substance. But the most crucial part is how this external matrix physically connects back to the cell's internal skeleton. And it does that using a chain of special connector proteins that actually span the cell membrane. And this is the key right here, this direct chain of connection. The internal cytoskeleton is physically linked right through the cell membrane to the external matrix. This creates a direct communication line, allowing events and forces in the outside environment to influence the cell's internal state and its behavior. It's a true inside-out integration. So let's bring it all home. We've journeyed from the cell's internal support system, the cytoskeleton, all the way out to the complex structures it builds to interact with the world around it. This table really drives home the fundamental differences. The plant cell wall, built from cellulose, is rigid and primarily protective. In stark contrast, the animal ECM is flexible and plays a much more dynamic role in things like cell adhesion, support, and communication. And that leaves us with a really powerful final question to think about. How did these fundamental differences at the cellular level, a rigid box versus a flexible web, give rise to two vastly different kingdoms of life, one stationary and rooted, the other mobile and dynamic? It's just a fascinating example of how structure truly dictates function, from the microscopic scale all the way up to the entire macroscopic world we see around us.